Khurana, who's actually starting our seminar series for this fall. She's an assistant professor at Weill Medical College of Cornell University. Associate professor, it's changed since the CV. Congrats. Great. And an affiliate member of the New York Genome Center. So some of you might be a familiar face, did a postdoctoral fellowship here at Yale with Mark Gerstein, and did her PhD training at the University of Pennsylvania with Michael Klein. And today she's going to tell some very interesting things about cancer and the non-coding genome. Thanks.
Thank you. Thanks so much, Steve, for the invitation. And thanks to all of you for coming. I'm going to say the cliche, but it's great to be back in person and talk to everyone and be here. And yeah, so as Steve mentioned, I was actually at Yale in Mark's lab, and I see at least somebody from Mark's lab here. That's so very excited to be back in New Haven. And today I'm going to talk to you about cancer evolution through the lens of the non-coding genome. So I learned about the non-coding genome when I was in Mark's lab, and I started applying it on cancer when I moved to the Cancer Center at Cornell and at the Bayer Cancer Center at Weill Cornell and Memorial Sloan Kettering Cancer Center. And you will hear our, our journey through that. So, so the questions that I'll be, discussing today, the main themes of the work that I'll be discussing, one of them is which genetic changes drive initial tumor growth. And obviously, most of the effort in this direction has been on protein coding genes. And in one of the recent efforts, we participated and ran our methods along with many other methods in the TCGA project. We created this catalog of the protein coding cancer genes by analyzing about 10,000 TCGA tumors. But this was focused on protein coding genes. And actually, at least 25% patients do not show mutations in known protein coding drivers. And even when they show, you usually need more than one hit to drive cancer cells. So the question that we try to answer is, are there genetic changes in the non-coding regions of the genomes, like gene promoters, gene enhancers, CDCF insulators that can have a role in tumor growth? And then the other big question that we try to address is the genetic and the epigenetic changes as tumors evolve resistance to drugs. So for those of you who don't work on cancer, might not be familiar, but the entire TCGA and ICGC data is just primary cancers, except melanoma, where we have some metastatic genomes. But the effort is focused completely on primary cancers. So there is this entire challenging world after primary cancers. Like once you treat the cancer, actually, in most cases, we wish it was like this, where the drugs would have solved all the problems, but it's not like that. The reality is that the cancers escape the drugs and they evolve resistance. And this is a big challenge in prostate cancer, breast cancer, liver cancer, many other cancers. And how do we understand what's going on, and can we use our knowledge of the non-coding genome to identify potentially novel drug targets when cancers evade drugs that are given as a first-line therapy. So these are the key questions that we focus in the lab, and we use our expertise in the non-coding regions to answer these questions. So in the first part of my talk, I will discuss about the genetic changes. And then the second part, I'll talk more about treatment resistance in prostate cancer and how we identified novel subtypes by looking at the chromatin and epigenetic changes. There's a lot of work that I'll trying to include here, and we can make it more interactive. And I realize some of the work, like on structural variations, that you guys might be interested in. I didn't include slides and the CRISPR screen, but we can discuss that as we go along. So over the years, we have developed some methods to basically predict the functional impact of non-coding variants. And my journey into this started, like I said, when I was in Mark's lab back in 2013. And then this method that we developed was called FunSeq because it was so much fun. But it was for the functional impact of sequence variants. And there we use information from conservation across species and across 1,000 genomes to basically identify regions that are under negative selection. And we use multiple other features that also showed trends of negative selection, and then came up with this approach to score mutations, which was an entropy-based method, and use allele frequencies from 1,000 genomes population to score mutations based on their potential deleterious impact. Then some of the other methods we have developed over the years is to predict loss of function tolerance of enhancers. And this was primarily led by Duo, who is a postdoc in my lab. And here what we did is we used known loss of function tolerant enhancers. What that means is the enhancers that have been lost homozygously in healthy individuals. And these individuals don't have an obvious phenotypic deleterious impact. And then we use some low loss of function tolerant enhancers, which is the enhancers that have been annotated to be essential, and their removal has shown a very strong phenotypic impact. So, and then we train machine learning models to using networks. I'm going to talk about that a little bit as we go further, to basically predict what would be the loss of function tolerance score of a given enhancer sequence. And then one other method we developed is called DeepMILO. This was developed by Tuan in my lab, who is now working with Facebook. So found greener pastures, I guess. But so here he used a deep learning model to predict impact of variants on chromatin structure. So basically what he did is, actually I can go to the next slide here. He used data from CDCF ChIP-seq experiment. So the ChIP-seq assays, those of you, in case you don't know, they capture where the transcription factor is bound and which regions are in contact with that region. So he used the anchor regions from the CDCF ChIP-seq assays from multiple cell lines to train the model on the insulator loops and non-loops. And the idea is that once we have this model, which was a hybrid convolutional and recurrent neural network, you can input your patient's variants, either structural variants, single nucleotide variants, or indels, and then it will tell you what is the probability that the the loop was there and has been perturbed in the presence of the sequence variants, and that's how you can infer the impact of sequence variant. And you can either input a default set of insulator loops that's available from ENCODE or Roadmap or wherever you got it from, or if you do a cell type specific or cancer type specific ChIP-seq assay, you can also input that and then predict the effect of mutations on those loops. So just a little bit more about what I was talking about, predicting the loss of function tolerance scores for enhancers. So this Duo basically used something that we called a meganet. So when I was in Mark's lab, I developed multinet, which was all sorts of protein interactions. And then Duo was like, okay, we're going to go mega now. We're going to include gene regulatory interactions in this. So we developed meganet, which includes the enhancer to gene and gene to gene interactions. And then we used the network features of this. And then those were the features basically in the model. And like I was telling before, that the true loss of function tolerant and the low loss of function tolerant enhancers were the gold standards. And as you can see, we can actually get a pretty good AUROC on these. And turns out that the features of the genes that these enhancers target are very important in predicting the loss of function tolerance scores of enhancers. And so these were, some examples of methods that we have developed to predict the loss of, to predict the functional impact of variants. And as you all probably know, that there are many methods in the community to do that, right? So, but when we specifically talk about cancer, there's one other challenge that we have to keep in mind, which is that the somatic mutations are not, they do not arise randomly they do not arise uniformly in the genome. So, so naively, when people want to identify cancer drivers, what's the signal that they most commonly use? It's the frequency of the mutation. Is it how frequent is it in the patient population? And obviously, it can tell you a lot. But we have to keep in mind that some regions can be observed to be highly mutated in patients, even though it's not because of positive selection. And one of the covariates of these mutation rates is actually very strongly observed in melanoma and lung cancer, where this was revealed in a couple of papers in 2016. That what happens is that when transcription factors are bound to the DNA, they kind of prevent the nucleotide excision repair proteins from binding, which is the main mechanism of fixing the lesions caused by UV or tobacco smoke. And as a result, you see a higher mutational density at transcription factor binding sites in melanoma and lung cancer. And if you're not aware of this and you develop model to which regions show higher mutational density, and if the assumption is that it's all because of positive selection, you're going to identify many drivers. You're going to say these are all drivers. And believe it or not, I have reviewed papers for very high impact journals where authors were adamant that they identified 200 drivers or so, right? So these are the kind of things that we have to keep in mind when we are trying to identify regions under positive selection in cancer. And in fact, it was beautifully shown by Polak and others in 2015 that if you use the histone modifications from the original, cell of origin. So if you're looking at melanoma, if you use the histone modifications from skin and so on, you can explain a lot of variance in the mutation rate, meaning it's the underlying covariates that are giving rise to this heterogeneity. And this is what we have to keep in mind in our statistical models when we are trying to identify drivers. So with that in mind, we developed this approach called CNC driver, which was, we said Cornell Non-Coding driver because NC driver was already taken. So for detecting driver coding and non-coding elements. And this was developed by Eric, who was a graduate student in my lab and is now working at Memorial Sloan Kettering. So, the idea behind this approach was that it was a non-parametric approach where we analyze the mutations in the functional elements, either the promoters, enhancers, it's also for the coding genes and the link RNAs, or the CDCF insulator boundaries. And then we predict the functional impact of these mutations, which we can do using the approaches we have developed. Or if you want to run this, you could use your own approach. And what we do is we compute the score, which is called the composite score, which integrates the frequency of the mutations and the functional impact. And then the null model is created by sampling the same number of mutations from the same element type in the same cancer. So if you're talking about melanoma enhancers, we would create the null model from melanoma enhancers. So to account for this covariates that would change the mutational density at melanoma enhancers specifically. So our way to, and then of course we can calculate p-values for if the score is significantly higher, and then we can check if we have actually, if the model is well calibrated with the QQ plots. And this is just a schematic, but basically then we can run this method on all functional elements or any cancer type and identify the putative or predicted drivers or predicted elements with more functional mutations and predicted by chance to be candidates for positive selection. So we have run this on genomes that are available from ICGC, TCGA mostly, and we were members of the PCOG consortium that many of you might have heard about, which was before COVID, so it was great. We actually traveled to a lot of these countries on the map here. So, it had cancer samples from patients from many different countries. It was a collaboration between ICGC and TCGA. And it was kind of the first large-scale effort to identify to basically analyze whole genome sequence data and identify drivers. And actually, an anecdote related to that: when I was in Mark's lab, I remember we had 100 prostate cancer genomes or so in 2013. From TCG, and we could not identify mutations from those genomes. And we looked everywhere, and turns out nobody had done mutation calling because the cancer community didn't know what to do with whole genome data. So we then identified mutations ourselves. But this was basically the PCOG was started around the same time in 2014 when a lot of cancer whole genomes became available. And it was a big question how to analyze this data. So I am going to show some results, our results of running CNC driver on the different non-coding elements on the ICGC and TCGA data. The concerted, the integrated PCOG effort obviously used many methods, but the results were more or less similar. So, so sorry about the many QQ plots, but basically it's to give you an idea that how many candidates we identified in which different cancer types. So we identified at the gene promoters, we identified 20 candidates in 13 cancer types. And the only one that was found in multiple cancer types was the TERT promoter. No surprises there. This was the biggest, non-coding region that got the cancer community excited about mutations at regulatory regions. And then for the enhancers, we only identified 5 candidates in 7 cancer types. And they were, I think we discussed 1 or 2 of those in more detail in the PCOG paper. One was for TP53, but there was no, it was not like tons and tons of enhancers are showing positive selection. And from the link RNAs, long non-coding RNAs, we identified 30 candidates in 18 cancer types. And the one that was found in many different cancer types, I think that was NEAT1 and this one BALAT1, which as you can see is found as a hit in many different cancer types. But I think the jury is still out there. The debate is still ongoing if they're real drivers, because they seem to correspond to the mutational signatures that are found in these cancer types. So are they really functional or not is yet to be answered. The CTC of insulators was not part of the PCOG effort, but we identified candidates and insulators in my lab. And we've identified 21 significantly mutated or candidates for positive selection. And one of these was especially interesting because it seems to be in the vicinity, or it seemed to be in the vicinity of the TGF-beta gene, which is a very important gene in melanoma. And it promotes, tumor cell migration. And we saw that it was also associated with higher gene expression. So we went ahead and with collaborators at Weill Cornell, Effie Apostolo, and with Neville Sanjana at the New York Genome Center, we validated this with using CRISPR, and using the 3C assays, we saw that indeed the loops were, CTCF loops were as we had thought they should be. And then using CRISPR, when we made these 2 mutations, we saw that indeed the cell growth, we see an increase in cell growth here. So, so that was kind of, nice to see that in fact these mutations can drive the increase in cell growth and change gene expression. And I would, mention that many in the community besides PCOG, many different methods have been developed and run on different sequencing data sets, and we realized that there is really there is a need to put all this information together, because if you identify coding cancer drivers, you can go to COSMIC or many other cancer databases, but there's no real place where you can identify the non-coding drivers that have been identified. So Eric developed this database where we have put together all the papers that we know of and that keep coming up to identify non-coding drivers. So hopefully some of you will find this useful. We also put the evidence that has been used to identify those drivers, so you can see what's the confidence in those predictions. If you have, if you identify drivers, shoot us an email and we'll incorporate in this database. so, you know, like after the end of PCOG and the stage where we stand currently, we basically reached we are at a point where the people feel that there are not as many non-coding drivers as we would have thought in 2014. And is that really true, right? Are there no more non-coding drivers? Should we just give up whole genome sequencing of tumor whole genomes, just do exome sequencing? I'm sure you can guess which side I stand on based on the title of my talk. So I definitely think that there are still drivers out there to be identified. And one of the reasons we think that potentially we missed a lot of drivers is because of the underlying genomic annotations that were used to, I'd call, enhancers, for example. So the enhancer annotations from cancer samples and accurate enhancer to gene connections were missing in the entire PCOG effort, right? We used a set of reference annotations from ENCODE, but we know that they do not represent the diversity of the actual enhancers in patient samples. And I think that another big thing that was missing was some sort of functional validation and also high throughput CRISPR screens, which can tell us a lot more, you know, what are the, if you perturb the element, what does it do on the cell growth and gene expression? And this is something that we actually have exciting data on with Neville's lab at the New York Genome Center, but I'm not going to talk about today because of the constraints of time. I'm going to discuss one effort that we have developed to address the second bullet point, which is to improve, you know, the enhancer to gene links in cancer samples. So only actually in I think it was a couple of years ago, or maybe now 2 or 3 years ago, the Chromatin Accessibility data for in 2018, sorry. The Chromatin Accessibility data was published for 371 TCGA patients. So this really was, you know, the first paper that which we also participated in as part of TCGA, where the diversity of the open chromatin regions in all these cancer types was, you know, we started to find what are the annotations of the open chromatin regions in these cancer types across patient samples. So, and here you can see how many patients were sampled. So the most of the patients were actually breast cancer, colorectal cancer. And as you can see, this is still far from complete. You know, it's still a very small number of patients, but at least we got the first map. So the problem with when we are working with cancer patient samples, unlike working with cell lines, is that you don't have enough material to do all the assays that you can dream of when you think about the epigenome. So the histone modifications, the ChIP-seq, you just don't have enough sample, right? So what we did is, because the ATAC-seq data became available and we realized that now people can, you know, ATAC-seq technology can be used on patient samples. So we can get ATAC-seq and RNA-seq data. So we developed this approach which can actually integrate just ATAC-seq and RNA-seq to annotate potentially active versus poised elements, so we can predict which regions out of these open regions have H3K27 acetylation versus the H3K27 trimethylation. And we also predict the enhancer to gene connections using this approach. So I'm going to just walk you through this overall, you know, what this method does, which is called actually DGTAC, which I didn't put here, but it's because it was like ATAC, DGTAC, like differential gene targets of accessible chromatin. So the gene targets can vary in a single patient. And what the correlation-based methods, which was done in the science paper, they just assume that the same enhancer to gene link is present across all the patients because they just correlate, right? But these connections can actually be patient specific when the enhancer, for example, when the enhancer is in a poised state in a specific patient. So, so again, Duo and Andre, who was a grad student in my lab, and he's also working at Memorial Sloan Kettering now. They developed it, and the basic idea is that first they took the data from all these 371 patients and developed a regression model to collect the joint effect of the neighboring peaks, the open chromatin regions, on the expression of each gene. And then in each patient, they contributed they estimated the contribution of each peak to the gene expression. So you can think about it as the residual or the error term. And actually, sorry, this was the trained using this. I should probably have mentioned it first here. That this was trained using the high ChIP and ChIP data. So for the breast cancer, we had the high ChIP data, and we knew what were the for the ER positive MCF7, what were the contacts from the high ChIP data, what were the loops. And then they use many different features, including the expression of the gene, the peak signal of the ataxic peak signal, the distance to TSS, this residual or error term, the copy number alteration of the gene, etc., as the features to basically predict which enhancers are connected to which genes in a patient-specific manner. And I'm not going to go through all the details, but just to show you that when the regions where the open chromatin region was predicted to be connected to the target gene in a patient, we said these are the candidate active regions, so they should show an enrichment of H3K27 acetylation. And here is analyzing this from all the ENCODE cell lines. The H3K27 acetylation, as you can see, is indeed enriched to open regions where we predicted the targets relative to the ones where the target gene was not predicted, even though they're open, the chromatin is open there. And similarly, we see that the regions predicted to be in a poised state, we see enrichment of H3K27 trimethylation, again, all the cell lines from ENCODE and the data from ENCODE to validate this. So this kind of gave us, some confidence that we are indeed predicting the enhancers and to their genes correctly in a given patient sample. And of course, we identified some cancer type specific enhancer to gene links, and the enhancers that regulate cancer genes are obviously of special interest. And here you can see in this bar, so these are the different cancer types, and these are the different links. You're not supposed to read all of them, but you can see in this bar that actually, so the dark blue lines indicate where the correlation-based method could identify. And you can see that many of them are indeed the correlation-based method missed. So we identified these enhancer to gene links that could not be identified in the earlier work. And one of them was very interesting to us in breast cancer. These two enhancers that regulate ESR1, because we found that it's really specific to the luminal A and luminal B breast cancer samples, as you can see here. These are the two enhancers, and this is the predicted connections. So the orange samples here are the luminal samples where ESR1 is expressed. And the gene expression is higher in these samples. And indeed, it cannot be, before this identifying these enhancers, a copy number amplification definitely does not explain the higher ESR1 gene expression in the luminal A and B samples. And but these enhancers clearly, are present specifically in these samples. And then we also performed CRISPR-I validation. And indeed, if you, inhibit these enhancers, you see that the expression of the gene goes down. This is showing the effect of silencing of both the predicted enhancers relative to the promoters. So, so we hope this tool can be used to identify enhancer to gene connections just in the presence of ataxic and RNA-seq data from patient samples. And we also validated this with the ABC model, which is very popular, and I'm not showing those results here, but ABC model actually also needs H3K27 acetylation, and in fact, even high C, though that can be substituted for distance, but this can be run just using ataxic and RNA-seq. Okay, so I'm going to switch gears and talk about the, go to the second half of my talk and talk about not about primary tumors, but resistance, treatment resistance in metastatic cancer, and specifically I'm talking about prostate cancer. So lineage plasticity, which is kind of, we think about it as when the cells do not depend on the same transcription factors or master regulators that they used to depend on. That's how we think about lineage plasticity. It can have different definitions. So the resistance mechanism and lineage plasticity is a big problem in prostate cancer. So initially, all prostate cancer, when the patients show in the clinic, all of them depend on androgen receptor, which is critical for the development of prostate cancer, and which is why enzalutamide and abiraterone, they constitute actually the first line of therapy is castration, and then enzalutamide and abiraterone, which is the AR inhibitors, they are used. But resistance almost always emerges. And it could be still dependent on androgen receptor, for example, if there are mutations in the drug binding sites. And in fact, yes, that's observed. But it can be AR independent. And something that the clinicians knew always is they can actually see that the cancer looks different. It doesn't look adenocarcinoma, it looks neuroendocrine, which is like pathology. You can see that it looks different. But then there are also these tumors where they don't look neuroendocrine and they don't depend on androgen receptor. It's kind of somewhere in the middle, and they're called the double negatives in the field. And this remained to be like an open question. So what did we do? We basically worked with a clinician across the street at Memorial Sloan Kettering UTEN, who is a prostate cancer oncologist and sees these patients. And what he does is he develops these organoid models from patient samples. And we also have some patient-derived xenografts. So we were like, okay, let's do the things we have been doing with cell lines and primary cancers on metastatic prostate cancer. So because the main motivation for this was because the traditional cell line models are very limited and they do not capture the diversity of patients. So not much can be learned by looking at the 4 cell lines that are available in the community. And the DNA sequencing showed that there are no clear, so the DNA sequencing was performed, mostly exome, but a little bit whole genome also. But that showed that no clear genetic alterations that could explain this lineage plasticity. We saw there were enrichments of some genes deleted to be in the lineage plastic tumors, but it was not mutually exclusive. And of course, it needs to be, it's important to identify, doesn't need to be stressed enough that it's important to identify the drivers or the dependencies of these tumors, because that's how we can target them. So coming with a background of regulatory network construction using chromatin accessibility and gene expression data, which, like I said, we did a lot with cell lines, and also from my lab, Priyanka did that. She was a postdoc and she did that for primary prostate cancer back in 2017. And we had identified some really interesting genes, which was an ERF deletion that was then reported in 2 other papers in the same year, using this approach that as prostate cancer drivers. So we were like, okay, so let's use these same techniques for castration-resistant prostate cancer. So the first thing we did was we generated ATAC-seq data for these 40 samples, which were organoid models, patient-derived xenografts, and then the cell lines that are available in the community. Yeah, it's marked here, which were the different samples. You can see that here. So there were the cell lines that were available. So this work was done by Fan Yang, who was a PhD student in my lab and is now working at AbbVie. So, so the first thing that Fan Yang saw was that, well, it's very clear that we get these 4 different clusters of the samples. And she used many different methods, and this seemed to be robust. And as you can see by the legend here, one of these groups is actually the AR high group, which is what we expected. They still rely on androgen receptor. And then this is the group that is the, actually, that's the mystery that gets solved in the next slide. So one of these groups that, so we were like, okay, these are the 4 groups, but how do we actually identify what do they correspond to based on what we already know? And that's why we looked at the gene expression. So besides the ATAC-seq generated RNA-seq data, and we looked at the gene expression in these groups, and it was very clear that this AR group was, enriched in the AR genes, AR pathway genes, and then there was this neuroendocrine group that was enriched in the known neuroendocrine genes. And then there were these 2 groups, right, that are not neuroendocrine, not dependent on AR. And one of them was clearly enriched in the Wnt signaling pathway. And one of them was actually enriched in many different pathways, which we are calling here, we ended up calling it stem cell-like because it was also enriched in the stem cell genes. But as you can see here, in fact, there's, JAK-STAT, and there's like the inflammatory signature, there's the FGF signaling, it's like really enriched in many different pathways. So which actually tells us why the drivers of this group were not identified before we did ATAC-seq. Because if you just do RNA-seq, even on patient population, you just get so many pathways that are enriched. And there are no clear genetic alterations that identify what is driving this, which is where the ATAC-seq data became super useful. And I have to say that after our paper came out, I think this was in May, subsequently, I think a month ago, Charles Sawyers published in Science, again, validating that indeed they see this group with single cell analysis, Charles Sawyers and Dana Pear's lab, where they emphasize on the JAK-STAT kinase, but we think it's basically the same group and can be targeted via these proteins. So, so this is again, showing you the data that genomic alterations could not clearly separate these 4 subtypes, not going into detail, but we saw the usual culprits we expected for the androgen receptor changes, but nothing that clearly separated these this group, which we call the SCL-like, which is the purple group here. So what did we do next? We created regulatory networks. So Fanying used the methods that we have developed in the lab to connect these regulatory, the ATAC-seq peaks with the gene expression from RNA-seq for all these 40 models that which we had sequenced, and she created these regulatory networks. And then, she basically ranked the transcription factors in each of these subtypes that we identified with the ATAC-seq data based on 3 different features. One of them was the out degree of the transcription factor. So how many genes does the transcription factor target? One of them was the difference in expression. So how higher is it expressed in one subtype relative to another? And then the chromatin accessibility. So do you see enrichment of motifs at the accessible regions in one subtype relative to others? And bingo, I mean, this worked perfectly for the groups that we knew of, like the AR group. You see AR, FOXA1, which is a known master regulator for this group, and the other, the FOX family genes, as we had expected in the AR subgroup. And even in neuroendocrine, the NeuroD1 and ASCL1, so the neuroendocrine prostate cancer is actually very similar to the squamous, small cell lung cancer. And these are the known important TFs there. And so this also kind of made a lot of sense. And for these groups, the Wnt pathway, the Wnt subgroup, she identified these transcription factors, which again made sense going in, with the consistent with the Wnt pathway. And for the stem cell-like group, she identified FOSL1, which is a member of the AP1 family of transcription factors, and other AP1 family transcription factors like Jun and FOS here, right? So this was very interesting to us. And now, obviously, the next question was, well, we have used these model systems to identify these 4 subtypes, but do we actually see these subtypes in the patient samples, and what is their prevalence? So what Fanyang did is she took data from the Stand Up to Cancer Consortium, which is metastatic prostate cancer patients. She took the RNA-seq data, and then she created a template of genes for each of these template set of genes for each of these subtypes from the organoids and the xenografts, and then used the nearest template prediction approach, which is basically looking at the cosine distance of the gene expression from the patient sample to your template gene signature, and then saw where, do the patient, which subtype does the patient most likely belongs to. And then using the random shuffling, you can even give a p-value and only use the ones where you get a reliable assignment. So so as you can see here that, she did this with Stand Up to Cancer patients, about, I think, 300 patients, and then 100 patients that were sequenced at Weill Cornell. And again, castration-resistant prostate cancer patients. And what we saw very interestingly is that this group that we call the stem cell-like group is actually present in a lot of patients. It's like 34% to 27%, right? They were assigned to this. And if you look at the scores, which have been the neuroendocrine score, the AR score, this is basically a set of genes that were developed in independent studies. And this is sort of a validation for our approach to assign patients. So the patients that fall in the AR have a high AR score. The patients that are neuroendocrine get a high neuroendocrine score. So that's how we know we are, assigning the patients correctly. And obviously, so the stem cell-like group was of very high interest. And then in collaboration with Yu's lab, we did a lot of further validation assays where, for example, we showed that if you knock out FOSL1, the cell growth decreases specifically in the organoid that belong to this subtype, the CRPC SCL, and not the CRPC AR. And then another, a big question was that, okay, we have identified these TFs, but can we think about this to actually target these tumors, right? Can we use these regulators that we have identified? And for those of you, again, who work in cancer, the AP1 proteins, the FOSL1, they actually work together with YAP-TAZ and TAD in many different cancers. This is like the downstream of the Hippo signaling pathway, and it's important in liver cancer, colorectal cancer. And Fanyang actually came up with the hypothesis that maybe it's the same in our data, because she saw that besides AP1, the TAD1 motifs were also enriched at the open chromatin regions in SCL. And then she, you looked at the YAP-TAZ target gene expression, and we saw an enrichment in this SCL, the CRPC SCL group. And then we also did ChIP-seq, and in fact saw a large overlap of the binding sites based on ChIP-seq. So kind of, giving us further evidence that this is kind of this is the model in which all these proteins work together to drive the growth of these stem cell-like tumors, which is shown to be, which is true in other cancers too. So, so some other bunch of experiments, I won't go through all of these, some of them, but just to make the reviewers happy. So, it's showing the same thing, they're all organoids, and that, if we knock down YAP-TAZ, the expression of the downstream genes goes down, and the cell growth is reduced. And more interestingly, this is what the reviewers wanted, but we think this actually helped our paper a lot, where we showed that the FOSL1 and YAP-TAZ knockdown actually does start to change chromatin accessibility at the CRPC SCL specific sites. So in fact, they act as the master regulator, specifically the FOSL1, because when we overexpress FOSL1 in link gap cells, which is a known prostate cancer cell line with high AR expression, we see that the accessibility at the CRPC SCL sites actually starts to go up. So that's this pink line. So that kind of tells us that this is like, the pioneering factor that is increasing the chromatin accessibility there. And then a bunch of other validations using patient data, we indeed see that the YAP-TAZ activity is higher in both the Stand Up to Cancer and Weill Cornell patients in this group. And very importantly, the CRPC SCL patients exhibit shorter time on the enzalutamide and abiraterone acetate treatment compared to the patients that belong in the CRPC AR group. So this is kind of showing us the clinical importance of this, identifying this subgroup. I'm going to, I'm going towards the end by just saying that, in fact, we hope we are having translational impact of analyzing the cancer genome through the non-coding lens, because in our paper, we showed that these drugs, we started by looking at chromatin accessibility and building these networks, but then we identified these targets. And in our paper, we showed 2 drugs, voriporfin, which targets YAPTADs, but it's kind of called a dirty drug because it's not very specific, and T5224, which targets AP1. We showed that it has, effect on cell growth in the CRPC SCL subtype, but these were like the tool compounds. And now we are, collaborators from companies like Novartis and Ikena have reached out to us because they are developing clinical grade TEAD inhibitors that they are testing out in other cancers, and they want to test in this subgroup of patients in castration-resistant prostate cancer. And actually inspired by this, again, my student Andre developed kind of this generalized approach where, he took the ATAC-seq and RNA-seq data from TCGA and totally inspired by this work that, let's create these regulatory networks, identify the key TFs, and then identify the, the genes that cooperate with these based on the DepMap data. And then which of these can be potentially targeted based on the drug interaction database. And he identified some of the known targets and then came up with these new candidates, which we think, this approach can lead to potentially new targets in these different cancer types. So, of course, the question is like, what's next, right? So in the field of non-coding genome in cancer, and just a sneak peek into, what we are excited about right now is, the problem of what are these regulatory regions in metastatic cancer remains. So we solved this problem in castration-resistant prostate cancer by analyzing organoid data, and we are doing that in breast cancer now, and in patient with using organoids and patient-derived xenografts. But the truth is that actually these organoids and xenografts are very hard to develop, right? You need like world-class expertise to develop these systems. Actually biology offers this wonderful opportunity. So I think some of you who work in this field of cancer biomarkers must be aware that the nucleus of you can use cell-free DNA to identify the nucleosomal footprints of tumor tissue by whole genome sequencing of cell-free DNA. So what's beautiful is that we do whole genome sequencing of cell-free DNA, but because the nucleosomal occupied regions show a higher coverage, they are not degraded by enzymes. And the nucleosomal depleted regions are degraded. They show a lower coverage. So you actually see a very nice signal of nucleosomal footprints in the whole genome sequence of cell-free DNA, right? So, and this is being used by other labs and other companies to develop biomarkers for specific genes. But what we are interested in is, can we actually decode the epigenome of the tumor tissue by processing this signal, by analyzing the whole genome sequencing data of cell-free DNA. So we are, and of course, this is this makes everybody, it makes us happy because plasma is easily available from patients, right? So we can get plasma from all the patients and then get an idea of what their tumor epigenome looks like. So with that, I have a lot of people to thank. I hope I got everyone here. So a lot of folks in my lab, most of them actually have moved on now, the ones that are highlighted. The ones that are not probably will be discussed next time, their work. So that, but they have contributed also to all these projects, and our collaborators at Weill Cornell, and a lot of collaborators at Memorial Sloan Kettering, which really has this big resource of samples and especially the clinical expertise, and Neville at the New York Genome Center for the CRISPR work, and now Ben and Matt at Princeton for the computational methods for cell-free DNA, and all the people who have